Yoshinori Osumi.
Yeah.
Welcome to Nobel Week Stockholm.
Yeah.
All Nobel laureates were asked to bring a thing, an artifact, to the Nobel Museum in Stockholm.
Yeah.
So, what did you bring?
Yeah, I brought these two things. Yeah, and I want to explain. Those are the gift from my colleagues. Last year I got this, and this is a miniature of 27 years ago of me, and I didn't know that was a secret that I got this one after the celebration party was finished. They gave me this quite small. It's funny.
It's very, very cute. And this is the way you do your research, sitting by the microscope?
Always. Yesterday I said probably the origin of my the first I found the phenomena, and for me that was the starting point of my work, and so I like microscope to use to see something, and I believe the discovery of some sort of a phenomena is the most important for biology. Still, I think so. So many my colleague knows that, so they gave me this miniature. This is my probably 27 years old. So I had a black beard, black hair.
Yeah, but it's very much like you.
Yeah, thank you. That's this one.
Yeah.
And those one is yesterday I told about the what is ATG gene, and this is a ATG12. It's a ubiquitin-like molecule bound to ATG5 and combined by ATG16 dimer. So this is the first we solved the structure of ATG12, 5, 16 molecules, and this one was also present from a colleague 2 years ago or something. And this is a ATG8, another ubiquitin-like molecule. This molecule, the homologue of this molecule in mammals is so-called LC3. That is very important molecule for autophagy research. So those I brought. Yeah. And otherwise, I don't have many any special things. So
this is lovely. I was thinking, when you were born in 1945 in Japan, it was kind of a rough time then.
Oh yes, yeah. But I do not remember because I was so young. So then when I was a young boy, Japanese recovered so quickly, and everything goes up. So my young age, or up to probably undergraduate student, everything is quite positive period. Then then we have now, because now we have some sort of the mature society, and we have many earthquakes right now, volcano explosion. So we feel everything is not so stable. No. Yeah, I never saw that kind of thing when I was young.
No. I was wondering, when you were a young boy, what did you picture yourself doing as a grown-up?
Yeah, probably I said, I have no talent to drawing or sports or something, and probably I had not so many choices. Yeah, my father was a professor in Kyushu University, and my parents probably expected I'm be the scientist. That's all for me.
Was a
it's not.
It's not that far-fetched for you.
Yeah.
But you started out studying chemistry, and then you switched to biology. Why was that?
Okay, a little bit I said in the lecture. You understand the chemistry is so a long story and also established. So when I went to the Tokyo University, chemistry lecture was not so stimulative for me. But that time, as I said, the molecular biology appeared, and everything is so fresh for me and it's so fascinating for me. So I want to be a molecular biologist. That's the reason I have no, for example, in high school age, I haven't taken a biology course lecture, but immediately I interested in the molecular biology at undergraduate school. Yeah.
So was it any particular person that inspired you?
Molecular biology?
Yeah.
Probably I won't say in the lecture, that was really a good time for me. So just every day were, for example, genetic code is differing. So ATG means methionine or that kind of things. It was so exciting for me. So that was, of course, I learned about the Nuremberg or Chor and something. Yeah, that's my age of their molecular biology. Yeah.
According to your experience, what personal characteristics are valuable to a scientist? What do you need to?
I believe it's not a stereotype of researcher. I'm not typical.
Why not?
I don't know. I like, as I said, I'm not a competitive person, and I want to study something I'm interested in. But many successful scientists do not behave like me. I believe, but I hope that not only scientists, our society should have heterogeneity. That's very important for human. Yeah, so
a lot of different people are needed.
Yeah, so even scientists, we should have many types of scientists, and I don't know what should be. I don't want to say this is the most typical or most important way to start science. Yeah, so you have. Yeah, that's my opinion for scientists. Yeah.
And you're still running a research group. You said that it's important with different kinds of scientists. Do you think like that when you're in your team? Do you also have different varieties?
So, of course, when I started autophagy, just the important thing is what's going on by morphology or EM study, and then genetic start, and then molecular biological research started. So even autophagy, we should focus some part of their subject during my 27 years. So, but I hope my lab has heterogeneous people just doing research to solve their autophagy. So someone should be the molecular biologists, some were geneticists, some were biochemists, good biochemists, some cell biologists. I want to have a quite heterogeneous people in my home. And also,
do you still do your own experiments?
Sometimes I try, but it's very difficult. Yeah, we need some continuing time. So, yeah, I started someday, but I should give up to continue. So it's I cannot say I'm doing, but I love research by myself. Yeah.
I was thinking if we turn to your discovery. Let's see. So this is, if you can say, this is, if you show it to the camera like that. That's a very schematic picture of what autophagy might be. But you show that autophagy, which is self-eating, it's not only a way of degrading material, it's also about recycling. So why does our cells need a system for recycling?
Okay. I always said, for example, if you not have any food, it's quite often a challenge in nature. For example, you can see many TV programs show there. Polar bear cannot get any food for several days, and many you can have that kind of even lion cannot get. So, in evolutionary, starvation is the most
it's a big challenge to the organism.
So we should have the recycling system at the beginning of our life. So I think, for example, if we one day starvation, I don't know, but I heard our liver shrink to 70% of the volume.
Wow.
Yeah, that means autophagy is very important for our whole body. Liver do the autophagy and degrade their own protein, and amino acid are supplied everywhere in our body, and especially for brain. And quite interestingly, if you starve our body, every cell, almost all part of our organ starts to autophagy, but only brain doesn't. So that means brain nerve system is so important. So first they are protected for starvation. That means there are other, for example, liver is so important for survival. Yeah, so I believe recycling is very important. Yeah, I want to emphasize in my lecture. Yeah.
And if you are a cell, too much autophagy or too little, what is the worst problem for a cell?
Probably, as I said, of course, so the recycling problem with, for example, our autophagy defective cell cannot survive under starvation condition. That means recycling is very important for survival. And also, as I said yesterday, autophagy has another function to get rid of the harmful excessive materials. So if you have defect in autophagy, many harmful protein or harmful or dangerous organ accumulate in our cell. That is also very dangerous for life. So autophagy defect, now we know autophagy defect has many defect in the health or disease problem. That many people interested in autophagy. That's one of the most important.
Yes, because if you talk about recycling and nature and the environment, there are some materials in our environment that are not easily degraded or recycled, like plastic things that end up in the oceans. Is it the same thing in cells? Are there particular materials that are hard to degrade and recycle?
Yeah, probably in usual case, in lysosome has so potent activity to degrade almost everything. So, but we know many lysosome disease that cannot degrade, for example, specific proteins or specific molecule. Those are all goes some sort of serious problem for health. We know, long time ago we know many lysosome disease, but we didn't have any autophagy disease. Probably autophagy is more fundamental function. So probably if you have a severe autophagy defect, they cannot survive. So we didn't have any specific disease for autophagy. That might, I think that might be the case in autophagy. Yeah.
Maybe because here this should be like in a mammalian cell, because this is a lysosome and it's encapsulating the things that should be, and here it's like, but you did your research, that's why we had this little picture also with your favorite organism, the yeast. So that's why we bought that one.
First I want to explain, the autophagy, most important event is how to sequester a portion of cytosol into autophagosome. But nothing was known about the molecular mechanism forming this kind of structure. So
that was the thing you were solving.
So we need probably we need quite good model system. And when I started, I was working on vacuole and not lysosome. We have only this kind of big rigid compartment, and I assume this might be homologous to the lysosome. So I want to know if this is really the rigid compartment, how these material come across the membrane. So I thought from the beginning we are doing some sort of membrane biology, but the beginning, my starting point was found this kind of structure inside the vacuole, and then electron microscopy of this process proved this is exactly the similar phenomena shown this. And of course, lysosome is much smaller than vacuole, but you can see autophagosome is formed too, and then fused with a lytic compartment, and hydrolytic enzyme attack the inner membrane structure. That is exactly the same phenomena we have.
So when you were looking like this, at what point did you understand that you had made a breakthrough discovery? What was the...
Probably, as I said, probably when I saw the this is an electron microscope, but I just saw this phenomena under light microscope, and I saw this structure moving around in the vacuole, and gradually this structure accumulate in the vacuole, and that was so. It looks to me it's a quite fascinating phenomena. So I was convinced I might have a really interesting
So
phenomena.
what was the feeling in your heart at that time?
That was a really probably most exciting time in my life.
You mentioned that before, but you said that one reason why you started to study autophagy was that you don't feel that you're a very competitive person, so you choose that kind of field. So I was just wondering, if you were starting your career today, what field would you look into then?
Many times I ask a similar question. I always say, probably if I'm now, I might not study autophagy because it's so popular and many people interested. Of course, autophagy is just a general word for many autophagy. You can analyze structure biology and cell biology in many ways, but probably it's so popular, so I want to make another subject to work on. But
like what is the most neglected field to this year?
I always say that I'm too old to think about that kind. And for me, I always say, even in East, we know about 30% of the mystery of the autophagy in East. Probably less than 30% in mammalian autophagy. So there are many things to analyze further. So in that case, I might start autophagy in some aspect of the autophagy. I don't know.
Being a researcher can sometimes be frustrating. As I understand. And did you, during those frustrating times, did you ever think of giving up, just quit and do something completely different?
I'm not so hurried to have a good result, so I have not so much pressure to do. Of course, one reason is autophagy is so new field and not so many people working in autophagy, so I was really happy. Everything is new for me. But for example, when we started the cloning of ATG genes, we had every genes are unknowable, and we haven't get any answer to what they are doing. So that was really frustrating time, and I didn't so much worried about. But many people said at that time, you always complain on the science meeting, we have so trouble on the ATG genes, ATG protein.
So for how long time did you endure without getting the results?
But soon, we found the ATG12 system, and within 2 or 3 years, we got many new findings in autophagy genes. So from them, everything is some sort of we should get information about the ATG protein and how they are working in the cell. So we had many problems to be solved. So we started exactly that way. But probably I'm a biologist and I want to know what is really autophagy doing in yeast. So I couldn't pursue that kind of question for a long time because I had so many things to do. So now I go back to that original questions now in my lab. So I want to working on what and when and how what our materials is degrading lysosome or vacuole. That's my question I have.
You mentioned that when you were little, your mother had tuberculosis, yes?
Yeah.
And one of the first things you got to know was about streptomycin.
Yeah.
But you never were interested in being a doctor?
Oh yes, I know. Yeah, probably. If I saw many very good doctors at that moment, probably I think the medicine is one of the most important field, but I don't know.
It wasn't.
That wasn't the case.
No.
Yeah.
You like to look at the stars and the nature.
Yeah.
You are now. I mean, you mentioned that you've been struggling yourself.
Yeah.
But now you are about to receive the Nobel Prize, and very successul. So, do you have any piece of advice for younger scientists that are now in the process where you were before? What?
Yeah, unfortunately, I saw the special in Japan, but I met Swedish scientists yesterday or the day before yesterday, and also they feel it is not so good time for basic research because a little bit younger generation in Japan, for example, professor 50 or 60, it is not easy to get the money, so they should write a grant application every time, and also it's not so easy, and they feel very competitive, and also many other administrative job things happen to them, so they have not good time to concentrate on science. So young generation watching their PI, they look not so happy when I was in a young generation. Yeah. So I hope I want to change that kind of situation.
In what way?
Yeah. And but for young generation, if you these days what happened for them is you should have a good paper when you have the undergraduate school or postdoc. You should always push to have a big journal paper or something. But it means if you should work on popular topics because it is much has a chance to get the Nature or Science or a Cell paper. But so it is very difficult to stick on the new subject. But
like you did.
Yeah. So my advice to the young generation: if you really have you can believe you can have good result in that many very competitive field, you let go. But if you really interested in something, you had better keep that question. Yeah. So don't hurry, or you had better find the phenomena you can continue 5 years, 10 years, 20 years.
27 years.
Yeah, yeah. That's my advice for the young generation.
Finally, we have talked a lot about your work and your career, but when it comes to life outside of the laboratory, what is important to you?
Yeah, probably in my case, my wife took care a lot of me, and I should appreciate her help. But yeah, it's very difficult to do everything so nicely. But now I want to have a more calm and more relaxed time. So it's a it's a hard question for me. Yeah.
I was just thinking, maybe have your family and your relations have been important for your scientific achievement?
Yeah. Yeah, probably I had 2 sons, but they didn't enter in the science field. Probably they watch me and I don't know.
Thank you very much
Thank
for
you.
taking your time. Domo arigato gozaimasu.